Nampak,
What
Dok,
can you?
nampak.
Okay. Right, that's great. Okay, I hope everyone can open their camera so that I can see you. So let's start our class today. Assalamualaikum warahmatullahi Bismillahirrahmanirrahim. Alhamdulillahi Rabbil Alamin. Wassalatu wassalamu ala ashrafil anbiya wal mursalin. Wa ala alihi wa sahbihi ajma'in. Subhanaka la ilma lana illa ma 'allamtana innaka antal alimul hakim. Rabbi shrah li sadri wa yassir li amri wahlul 'uqdatam min lisani yafqahu qawli amma ba'd. Nawaitu ta'alluma wa ta'lima wa naf'a wal intifa' wal ifadah wal istifadah wal hasa 'ala tamassuki bi kitabillahi wa bi sunnati rasulih sallallahu alaihi wasallam wa batigha wajhillah wa mardatihi wa qurbihi wa thawabih amma ba'd. So welcome back to the campus. I believe you have had one semester, right? In your first year on campus. Sempat ke Fahana?
The whole 1 year kita dekat campus, Doktor.
Oh, the whole 1 year. Okay. Okay. So, but the whole second year at home.
First semester only dekat second year, first semester. Betul kan?
Second year whole semester dekat rumah.
The
Okay.
whole semester, yeah. So it was a very long time at home. So welcome back. We had previously a class together, basic immunology, if I'm not mistaken, yeah. So our course is Hematology and Immunology Laboratory Practices. Here we assume that you have completed at least the basic and the basic immunology and basic hematology. Okay. It's not a required courses in those subjects. You can take this even not impose any prerequisite course to be completed before taking this course. Okay, so hematology and immunology laboratory practices, as the name implies, the focus would be on the lab part. Saya ni pun tak buka yang saya punya ni. Mana saya punya video ni? Okay, anyway, I will keep on talking first. Nanti saya akan tengok macam mana nak buka video. Okay, anyway, there are 4 of us teaching you. There might be some change because one lecturer may be added to help us deliver this course. But at the moment, there are 4 lecturers. You can see their contact numbers as well as email. Okay, if you need to contact us for any reason related to your academic, not restricted to this course. If you have problem with physiology, for example, genetic, and you want to talk to us, you feel more comfortable talking to us than the lecturers who are delivering those courses, you can also contact us. Myself, for example, apart from teaching hematology and immunology, I also I mean my actual training is in physiology, and I have a lot of training also in pharmacology, natural products, nano medicine. So you can ask me I mean questions not restricted only to this to the courses that on hematology and immunology. Okay, so this is a diagnostic course. Yes, about 11 of your friends are taking the research pathway. I believe you are aware that you can still change your pathways, kan? in the first few weeks, if you feel that you don't want to continue diagnostic, you want to go to research, you can still change in the first few weeks. So if what happened in the past, yeah, some students were not very sure about their pathway yet. Okay, they have been on diagnostic or research, but they want to make a final decision. In the first few weeks, they attended both lectures for the pathways, and there were cases where people change their pathways if there are slots available lah. You can request if you will want to change. There are also students who attended the lab. Here for Immuno lab practices ni kita ada lab. So some students from the from the research pathway, for example, they attended my lab, and I opened my lab for students from the research pathway. However, due to the restriction imposed on us in relation to the COVID-19, this year kalau ada kawan awak nak masuk, they need to tell me early on, because there is a limit to the number of students that could be in the lab, and also the limit with regards to the facilities that we have. Yang course macam ni, contohnya kita ada the mannequin to take blood. This year we found out that one or two of the mannequin, the arm mannequin, cannot be used anymore. So we are left with only 4. So that means we could only take up to 16 students to do the practicals. If any of your friends from the research pathway ask, then that they need to talk to me first. Yeah. Okay. So these are the skills that we hope you will learn in this course, communication skill. Okay. This is perhaps one of the courses where you will be taught on communicating, okay, in a clinical healthcare setting. In particular, when we are doing the phlebotomy or the blood collection, a very good communication skill is very much needed. You will see the emphasis there. Okay. We also hope that you will learn about critical thinking. This is diagnostic pathway, so it involves a lot of tests, and you are expected to learn how to analyze, how to interpret data. Okay. Most of the lab sessions will be on basic practicals. Okay, basic practicals, but they are very important practicals. They are the foundation, yeah, so to speak. We could not offer the much advanced practicals, yeah, because of constraint in terms of the finances, yeah. Sebab banyak yang lebih advanced tu dia guna more expensive materials. Tapi tak apa, you will be learning the basic ones. So when you get into the hospitals, you have a good footing already. Okay. The only thing that I ask from you when you are in the lab practicals is to give your all. Okay, you give your all. Maksudnya berusaha, ya, sehabis baik. Walaupun awalnya it appears difficult. Some of the basic practicals, they are basic. They appear to be easy, but when you try to do it in the first instance, it is quite challenging. You will see later on in the lab practicals. Yeah, we used to have very huge group. Satu kelas tu dalam 40 students, and some students because they are in groups, they don't take part actively. Yeah, dia duduk, dia tengok je kawan-kawan dia, and that was one of the reasons why we create this course, so that we can reduce the number of students that participate, yeah, and they can focus more. So one mannequin, for example, will have only 4 students on each mannequin. They have more time. They can be assessed much more properly by myself. So in my lab, I will walk from one bench to another, and I will ask you to demonstrate to me. We will be spending a lot of time doing that. Bukan bagi briefing and then leave it to you guys to do the practicals. No, my way of doing lab would be I will brief, I will demonstrate, and then I will let you do it first, and then I will come from bench to bench. Okay, everyone must demonstrate until or up to my satisfaction. Okay, kalau saya tak puas hati, buat dah kat situ sampai habis lab. After lab ke, tengoklah macam mana. Okay, so we have 3 hours, yeah, and that perhaps would be your only opportunity. So use it wisely, ya. Lepas tu nanti nak masuk dalam OSPI. Whatever I've taught in the class, ya, theory in the class, will be related to the labs and then will be included in the OSPI. Pernah buat OSPI sebelum ni?
Pernah masa year 1.
Masa year 1. Which class? Micro ke?
Betul.
Betul.
Okay. So so you have had experience doing OSPI, ya. OSPI ni dia pressure dia sebab time lah. You have very limited time. There's a lot of questions, a lot of tasks to be completed. But that would be one of the things that would help you developing your problem solving, critical thinking, and also lab scientific skills. So the OSPI is when you could shine individually. Kadang-kadang dalam lab you cannot show your real potential, but in the OSPI, that's where you can show your own ability to all of us. OSPI ni. Okay. So these are the topics. We have introduction to hematology and immunology system, laboratory system sebenarnya. Okay. What is this? Okay. Right. So our class is between 2 to 3 hours. Yeah. Dia tak hopefully I will not exceed 3 hours lah. Although if you leave it to me, may talk to 4 or 5 hours pun okay. Okay, then we have changed our timetable a little bit. So be careful. Do not refer to the timetable, the official timetable. Refer to the teaching plan. We have to make some changes because we want to do the OSPI as soon as possible. Year 4, current year 4, they still haven't done their OSPI. They have completed all other parts of the course except for OSPI because of the MCO. Masa nak buat OSPI je, masa itulah MCO tiba-tiba diumumkan. So they have to IP, we have to IP the subject, wait for another semester. So they will be doing the OSPI together with you on the same date this year. Hopefully, we will be able to do it lah for them. And I don't want the same thing to happen to you guys. I don't want you guys to be having IP for the subject as well. Nanti terlupa kan, some of the itu akan terlupa, and then nanti masa OSPI takut tak boleh nak perform. Sometimes it's not because you do not know, but because of the circumstances. So we want to we want to avoid that. Yeah, so we try to complete all the lab sessions as soon as possible, and then conduct the OSPE as soon as possible as well. Okay, now, so you need to be aware of the change in the timetable. Yeah, we'll be using the slot given to us on Tuesday as well as Thursdays. Yeah, so the Thursday in the official timetable, it is actually for lab use. Tapi kita tukar sikit lah mana-mana yang kita boleh pakai tu kita kadang kita guna untuk kelas. Likewise for the Tuesday, we will use it for lab session if it is possible. And then on Fridays, a few Fridays in the afternoon, because there is one of the time when you do not have any class, so we take the opportunity to conduct other classes. And I hope that because you can cross state these days, all of you please take note of the change and don't make any other arrangement. Ya, nak balik KL ke mana ke, nak balik ke Terengganu ke petang Jumaat ke, please baru je pun balik campus, I'm sure you can tak balik for some time kan, tunggu sampai habis. I don't want anyone to come to me and say that they won't be in class because they are going back to their kampung. Tell your mom, tell your parents, I've been at home for many months, so birthdays, get together, etc. can wait for a while until I've completed my OSPE. Cakap macam tu, okay? Saya rasa OSPE kita bulan berapa? Bulan 12. Lama lah juga kan. Tapi just 1-2 months kot. And our class is not every Friday pun. But just for your info lah. So please, I do not, I expect not to hear any request. Okay. Okay, any questions so far?
No.
So quizzes.
Doktor, saya ada soalan.
Ya.
Nadia. Weightage untuk subjek ni another 40% tu apa Sebab kalau kan ada dekat atas lagi.
Dekat atas. Final exam 30%. You have assignment 10%. Cukup kan?
Okay.
30, 40.
Okay.
Cukup kan?
Okay.
Quizzes ni very easy sebenarnya. Don't worry lah. Especially my quizzes, very easy. You can ask your seniors, what is the tips to answer my quizzes? Okay, very easy. Lab reports. Lab report ni memang senang sangat nak score lah. Especially if you attend the labs, 20 marks almost guaranteed. Takkan tak boleh dapat 10% at least. OSPE 20%. Within berapa? 2 hours you could get 20% dah. But dia kita bagi dia tinggi sikit because is the pressure involved, the challenge involved. So it's higher. Okay. So quizzes etc. will be in the later part of the semesters. We hope because quizzes are not very stressful, isn't it? Sikit-sikit je kan. We hope that selalunya hujung-hujung ni dia akan coincide with a lot of assignments, essays, etc. Right? So if you have any problem, you come to me and discuss. You can. We don't have to stick to what has been stipulated on the teaching plan. We can make amends to the teaching plan. Katalah on the same day as the quiz you have the assignment, you want to move it to another date, just talk to the individual lecturers. Okay, any question? All right. Okey. Okey, nampak saya sorry saya tak buka video tadi. So, berapa orang sekarang ni? 30. 30 termasuk saya. So, everyone is here. So, tak ada soalan ya? Can continue with our lecture. Okay. Saya pakai slide yang baru yang lama lagi. AHB 1103. I will change the code Oops. Sorry, anak saya baru bangun. Okey, iklan sekejap, sorry. Kita punya ni AHBS 3331 in case you are ni betul. Takut saya terbuka lecture note yang lainnya. AHBS okay. Ada yang tersalah kelas ke? Tak ada Right. Introduction to Hematology and Immunology Laboratory System. Okay. I've been teaching this class for the past 3 to 4 years. Sebelum ni tak ada. The laboratory part is part of basic immuno or basic hemato. So we have taken the laboratory components from both subjects and create another subject. So in this course, there are a few important things that you will learn. Firstly, a revision on the definitions of hematology and immunology and their interconnection. So if I ask you now, I hope you still remember what are the cells of the hematology system, the cells and organs of the immunology systems. You will remember that they come from the same pluripotent stem cell, and during their development, they change pathways. Some becomes the hematology cells, some become the immunology cells. Okay, and you will also learn in this particular lecture an introduction to hematology and immunology lab investigations, and then some introduction to the management of lab and the blood bank. This will be expanded later on in their individual lectures. Okay, so just a revision, what is hematology? Okay, hematology is the study of the form, cellular and non-cellular elements of blood. So we have cells in the hematology systems, erythrocyte chiefly, then we also have platelets and also the lymphocytes. Those are the cells of the hematology system. And then we also have non-cellular elements such as the plasma. So the plasma contains a lot of materials in it. It's very important. Then we have the importance of the hematology is because any alterations, especially in the form elements of blood, this is usually the result of disease. The results of disease. So if we look at the level of erythrocyte, the red blood cell, for example, if there is a change, this is indicating some changes, some pathological change, has happened, but they are often not the primary cause. They are often, they could be the primary cause as well, related to the cells, but they can lead us to other root causes. And variations in the form elements in the blood are often the first sign that disease is occurring in the body. And everyone who gets into hospital, they will get their blood taken for test, right? Other tests are carried out, but the blood test, like 100% of people who get into hospital will be having their blood taken. So that is reflecting the importance of the hematology system. Yeah, they indicate a lot of things, things that are abnormal to the patients, and also give us clues of as to what further investigations are needed. Okay, so the changes caused by these diseases may be detected by lab tests that measure deviations of the blood constituents from the normal values. So it is important for people who work in the hematology lab to commit to memory, yeah, what are the normal range for each type of blood cells, for example. You know, what is the erythrocyte level, and understand that different group of people have a different normal range. For men, it is different than the ladies. So ladies also different if they are in the state of pregnancy, and there is also a different in the normal range between adults as well as the kids, yeah, between people who are from different races. So Malaysia, for example, in the last 20 odd years, maybe not that many foreigners from abroad, from African countries, from especially, yeah. But these days, we in certain parts of KL, there are a lot of foreigners from Africa, and they have a different range of normality. So we need to understand that so that we know what are normal and what are abnormal. And then for immunology, immunology concerns with the study of how the body recognize self and differentiate it from non-self, yeah, self and non-self. And this is the basis of our immune system against invading pathogen, yeah. So it could be divided also into the study of the cellular component of immunology and the non-cellular components. If I ask Eva, is Eva here?
Yes, Doctor.
Ya, kenapa Eva tak duduk dalam natural product subject? Ingatkan ambil manuscript study, you be in natural product.
Tak tahu lagi.
Okay, I think it's good that you are here, because when you do the manuscript project, you'll be learning a lot on the plants, etc. You'll be learning a lot on natural products indirectly. So Eva, if I can ask, can you name some of these cells of the immunology?
B cell, T cell.
Okay. Anything else? So you have the lymphocytes, and then you also have the non-lymphocytes, and okay. So can you name some of the non-lymphocytic cells? Eva? Ingat, astagfirullah. Okay. Okay. I think it's good, yeah. Before you enter any of the classes, you need to get a book, yeah, in hand. Get it from the library and flick through, yeah, the reference book, and have some preparation, yeah, some preparation. That's how we could master a subject more easily, yeah. Dia kita liken learning ni, we cannot enter a class empty, totally empty, yeah. Kalau totally empty, the lecturer need to work very hard to fill it up, yeah, to the brim. But you cannot also enter a class totally full. Totally full ni maknanya macam you are the lecturer cannot help you anymore with regard to the learning. Yeah, dia normally dalam the philosophy of learning, we give this metaphora. Yeah, the student must not enter the class totally empty, because then it will be hard work to help the student, but also not to enter the class totally full. Totally full ni normally referring to the arrogant state of the student lah, do not want to learn, feel that they are already know a lot. Itu you need to be in the middle, half feel, yeah, have some preparation, so but still also have some spaces for the lecturers to fill you up, macam tu. Okay, so immunology is divided into the study of cellular component and the non-cellular components. Non-cellular components ni macam antibodies, yeah, is part of the plasma. Okay, kejap ya. I got some distraction. Kejap. Okay, tersekat maksudnya apa?
Tak dengar tadi, Doktor.
Ada tadi tu saya ada gangguan sekejap. We stop at the definition for immunology. Okay?
Okay, Doktor.
Kat sini ke? Kat sini kan?
Yes, Doktor.
Alright. Okay, tadi ada gangguan sekejap. Biasalah duduk kat rumah ni macam tu lah. Kejap anak lah. Okay. So itu ada dia punya pro and cons lah kalau dekat rumah ni. Kalau dekat dekat kuliah sekarang ni internet tak stabil. Dekat bilik saya tu tak stabil. So kat rumah saya lebih stabil internetnya, tapi dia ada banyak gangguan pula. Okay. Apa? Ya. Okay. So as I said in the beginning, the immune system is greatly connected with the immunology system because the origin of the cells. Yeah. So if the hematology system is affected, yeah, there are the disease, for example, the immunology system could also be affected. So macam kalau bone marrow, for example, yeah, the bone marrow, I mean anything that change the state of the bone marrow. Contoh orang tu ada chemotherapy, or the person has got some bone marrow disease, bone marrow cancer, for example. Both of the system will be affected. And then the way that the blood cells recognize each other, that will be also affected by how good the immune system is. Okay. Right, so this is the composition of the blood, and just to jog your mind. If you take a blood sample and put it in a vial, in a tube like this, and leave it either standing on the top bench or spin the blood sample around, sooner or later we'll get them to be separated into 3 layers. Yeah? So at the top part, we get plasma, and then there is a middle layer called Buffy coat, and then the top, the bottommost will be a red layer full with red blood cells. Okay, so in the buffy coat, this is where we will find the white blood cells and the platelets mainly concentrated. So the lymphocyte, granulocyte, monocyte will be there. We have techniques to isolate them, yeah, to isolate them, and then we from the blood sample that we take, we can produce blood products. Okay, kita panggil blood products. Okay, kita boleh ada different type of blood products. We could have whole blood. Whole blood means they have all this plasma, buffy coat, red blood cells semua. Macam bila when you donate your blood, you'll be giving your whole blood, and then this whole blood could be transfused to another person who lost blood due to accident and etc. So they have all this. But in certain cases, we want a blood product which contains only red blood cells. Sebab kalau kita dia ada plasma, they may have antibodies, etc., which can then attack the person who receiver of the blood. So kita boleh process the blood in such a way that we can take the plasma out, the buffy coat out, and leave only the red blood cell. So that is one of the product. In some cases, we want only the those components like fibrinogen only, for example. So we can isolate the plasma, take the plasma out into another tube, and then from there we process further until we only leave with the fibrinogen only, for example. And then we can transfuse in those who have problem with regard to the fibrinogen only blood product. Okay, but this is the composition of blood. You see some of these like neutrophils, for example, yeah, always being discussed under the topic of hematology, but it is also very important cells for the immune system. Yeah, for the innate immune system, the neutrophil is one of the most important because this is the most prevalent. Okay, kalau sekarang ni, if I take the blood from any of you, yeah, assuming that you are normal, you don't have disease, okay, of all the immune cell, the neutrophil will be highest in number, although you are healthy, yeah. Dia because dia in the state of preparation, in the state of dia macam standby against invading pathogen. Once any pathogen invade, the neutrophil will be acting first before the active immune system is being activated. Okay, now so these are the types of form element in the blood. Okay, can you name them? Masturina. Okay. Awak yang pertama saya panggil, tapi sebenarnya very advantageous I call you first because you can name me the most recognizable one. A
is red blood cell.
Yeah, A is red blood cell. So easy, yeah. How do you recognize A?
Because it doesn't have nucleus.
Sorry, can you speak louder?
Because it doesn't have any. It just have. It doesn't have nucleus.
Tak dengar.
It doesn't.
My question is, why do you say A is red blood cell?
Because it's clear.
What do you mean clear?
It doesn't.
Tak clear ke? Tak clear ke?
It doesn't have nucleus.
Yeah, tapi ada juga sel lain yang tak ada nukleus kan? What is?
It's shape. It's shape.
Okay, what is the other cell that do not have nucleus in the picture? J ada nukleus. Dia ada satu sel yang tidak di-label. Plasma. Did you say plasma?
Yes.
Plasma memang bukan sel.
Platelet, doktor.
Ya, platelet. Ya. Tapi ada ke platelet? Mungkin ada kot kat atas ni, mungkin platelet. Mungkin saya cakap. Okay. Anyway, so A is erythrocyte or red blood cell. Kenapa? Is very high in numbers. Yeah. So of all the cells of the hematology system, when we do a blood smear like this, yeah, this is a blood smear like this. We should be able to see a lot of red blood cell. Satu, they are the highest in terms of number. They are well-rounded, yeah, well-rounded. Although ada some yang not too round, but in general they are well-rounded. They appear red in color. So kalau yang ini tiba-tiba warna biru, it could be that it is not red blood cell or something lah. You may be using a different type of dye, for example, but red blood cell, most of the Romanowski stain that we use in the lab, they would produce red color in the red blood cell. It does not have a nucleus, that's correct. Okay. But more importantly, dia punya kat tengah-tengah ni, the area of pallor kita panggil, which is paler than the other area of the cell, is rounded and it is central. There is a central area of pallor. Okay. Macam kalau kita tengok yang lain-lain ni, if you look at the nucleus is not rounded kan. Tapi the red blood cell, area yang tak ada nucleus tu dia rounded and is pale. So those are the things that I'll be asking you while we do the lab practicals. Saya akan suruh awak buat lab smear. I will ask you to do this kind of lab smear. Macam mana nak buat? Kita bagi awak satu sampel blood, a few ml, and then you create this blood smear, stain it, look under the microscope, and then explain to me. Have you done any blood smear?
No.
Sasha tak pernah buat, Sasha?
I don't think so.
Tak pernah buat. Okay. Dia sebenarnya dalam sepanjang kos biomed, dalam kelas fisiologi, I'm not sure whether Dr. Radia have conducted the lab session with you to do blood smear. And also in class, in the class of parasitology. Dah parasitologi dah buat belum? With Dr. Azida.
The online session.
Online session only.
Online session. Itu the disadvantage of COVID lah. Otherwise, this would be your third opportunities to do a blood smear, and then do the differential diagnosis. Kalau macam dalam kelas Dr. Azida, normally dia akan suruh tengok dalam red blood cell tu awak boleh nampak malaria parasite tu. The falciparum, plasmodium vivax, semua you can see, and then you can differentiate whether they are mature or not, what stage of the infection is, so dalam kelas saya, kalau we happen to have example for malaria, very good lah, but normally we take sample from our lab staff or from myself or from you, we ask volunteers from the students to give blood. We take their blood sample just before the class start. Maybe that would also happen this year. And then they are all normal lah. Tak adalah yang ada malaria pula. Tapi kalau ada malaria, good for you lah, because we can detect it for free in the lab session. Tapi yang selalunya kita ambil macam itulah, dia tak adalah apa-apa abnormalities. Kecuali kalau dia baru demam. One of the classes we took the sample of the lab assistant who have just had a fever the day before. So the neutrophils were a little bit high than normal. So adalah some finding juga tu. Okay, otherwise it will be a normal blood smear, and then we ask you to count the numbers, and then you will see lah how many neutrophils, how many lymphocytes, how to determine whether B is lymphocyte or not, D is lymphocyte or not. E ni kenapa tengok lobe dia ada 3 lobe. Yang ni lobe dia ada berapa? 2 lobes. Yang ni lobe dia ada ni, but masked by granules. You can see the granules there. Ni pun ada granules, ni pun ada granules. Ni granules dia ada tapi the color is orange. Ada D ni. Some cells there is no granules, and then the lobe is almost of similar size of the cell, of the total size of the cell, macam the whole cytoplasm is made up of the nucleus. So saya kalau tanya Sasha tahu ni H ni apa ni? Can you name it, Sasha? Guess. Make a guess. What do you think?
Natural killer cell ke doktor?
Natural killer cell. Why do you think it's natural killer cell?
Because it's macam has a big
It's okay. Dia satu orang kata rule of thumb is what is common is common. What is common? Oh, ni ada ni platelet ni dia label G. What is common is common. So kalau yang lobe dia macam ni, dia almost completely filling up the cytoplasm. Perhaps the most common cell which present like this is the lymphocytes. Okay, lobe dia sangat dia punya nukleus sangat besar. Okay. Okay, so if you are not able or you have difficulties recognizing the cells that I put up on the slide, okay, I think you need to get a good book and have a read, Kenapa macam J ni? Ini kalau kita yang ditunjukkan dekat slide ni adalah common presentation of each type of cell. Common presentation, nanti kalau macam neutrophil for example, macam neutrophil ni kan dia nampak 3 lobe kan. Nanti I have some slide later on to show you the variation of neutrophils. Walaupun kita kata neutrophils is normally 3 lobes or more, but there are variations to it as well. So kita nak at the beginning ni for you to recognize what is normal. What is normal and what is common is common. Okay, remember my kata-kata hikmat ni. What is normal is normal. So you need to recognize lah normal tu apa. But what is normal. Jangan risau nak cakap itu adalah normal. Okay, itu satu. Kedua, what is common is common. Okay, jangan risau nampak banyak tu dia memang common. Dia common lah. And disease state tu kalau dia memang commonly like that, itulah dia. It will not change. Ni semua yang awak belajar ni adalah sunnatullah. Is the rule, is the system, is the syarat atau condition that Allah has set up in the world. And the rule, the law is almost always constant. Sama aja. So when it is very helpful once we recognize what is the law of God yang Allah dah letak, and then when we are presented with this kind of blood sample, we know whether it is disease state or not. Okay, right. So just to revise on the hematopoiesis, hematopoiesis or the formation and development of blood cells. It's very important because cells of the blood are constantly being lost or destroyed because of disease state, for example. And to maintain homeostasis, system must have the capacity for self-renewal. So this involves proliferation of the progeny stem cells, differentiation and maturation of the stem cells into the functional cellular elements. And these are some of the processes that affect the cells. Kita belajar yang common, yang normal, and then we also try to understand also that some variations could take place. Some of these variations happens because of this, because of the development of the cells. Contoh macam neutrophils, neutrophils. You will find the early neutrophils, they don't have more than 3 lobes, but they are neutrophils and they are normal. Okay, they are normal because it is developing. Okay, there are also cells, the neutrophil cells, that are more than 6 lobes, and they are already in the later stage of their development. Kita kena understand that there are also variation. So when you look at the blood sample, we need to know the age of the person, whether it is kid, adult, or the elderly. So, and then we look at the blood sample, we should expect to see that most of the neutrophils, macam mana, katalah in the elderly, most of the neutrophils should be normal, but we expect to see maybe about 5% aging neutrophils. Dan dia bukanlah disease state, it's just a normal presentation, macam tu. Okay. So we need to understand that there are proliferation, differentiation, and maturation processes taking place. In normal adults, the proliferation, differentiation, and maturation of the hematopoietic cells is limited to the bone marrow and the widespread lymphatic system, and only mature cells are released into the peripheral blood. Okay. So hematopoiesis begin as early as the 19th day after fertilization, initially in the yolk sac of the embryo. Okay, and at this stage only erythrocytes are made. Later on, they change from expressing fetal hemoglobin to expressing, when they are born, they move towards expressing adult hemoglobin. Dan ini more advanced stage lah. We will not do lab practicals on this. But if you take examples of blood from the fetus, we can run tests and they will produce different results on the in the test. So about 6 weeks of gestation, yolk sac production of erythrocyte decreases and production of RBCs in the human embryo itself begins. Okay, so the fetal liver becomes the chief site of blood cell production, taking over from the yolk sac. So erythrocytes are produced, and then the leukocyte and the thrombocyte production start to occur. Spleen, kidney, thymus, and lymph nodes at this stage serve as the minor sites of blood cell production. Lymph nodes will continue as an important site of lymphopoiesis, or the production of lymphocytes, throughout life. But blood production in other areas decreases and finally stop as the bone marrow becomes the primary site of hematopoiesis at about 6 months of gestation and continues throughout life. Okay, this is very important to understand. In some disease state like thalassemia, they are supposed to shut down the hematopoietic areas, like in the skull, for example. Dia patutnya dia berhenti, tapi because the bone marrow cannot produce normal red blood cell, the body in trying to fulfill the need of the body, dia akan buka semula all other areas of hematopoiesis. So the short bone, the skull will be asked to keep on producing red blood cells. And that's why in these people, thalassaemic people, for example, we see their skull enlarged. Enlarged. Kalau kita tengok dari tepi, dia ke depan, melonjong. Dia jadi macam jendul. Okay, and we see the expression of the red blood cell is also abnormal. So we make the link between the physical appearance and what we read from the blood sample. So most of you will be in the lab, receiving the blood sample. You will not see the patient. Okay, you will not see the patient, but you need to know the theory. So that bila kita tengok blood sample, the result of the blood sample tu kita tengok, this is indicating of thalassemia, for example. We can imagine the kind of person that is involved, and then why it happened like that, why it happened. So ini kalau kita diagnostic, kalau research, and then you take samples from the population, for example, studying thalassemia, etc. The blood sample, the result of the blood sample can conjure up, what actually happens outside the lab, what kind of patients have that present with the lab results, and what caused the results. Okay, so when the bone marrow becomes the chief site of hematopoiesis, leukocyte and thrombocyte production become more prominent. Okay, so anything that affect the bone marrow will affect all three lines of cells, the erythrocytes, and then the immune cells, as well as the platelet. So bila tu dia ada red blood cells, kemudian platelet ni untuk if you have wound, etc., you need to stop the bleeding. Itu the importance of the platelet. And then you have the immune cells. So your immune system will also be affected. People who have bone marrow, they will be prone to infection because of this. Okay, so this is the hematopoiesis graph showing the different part that will be involved in the production of the blood cells at different time point in life. So these dotted lines, this is at birth. So you see at birth, liver and spleen, the yolk sac is no longer producing any blood cells. The distal long bones also, they will start to stop producing, and as we age, only the axial skeleton will be kept as the factory for producing the hematology cells. Okay, this axial skeleton, axial means the central, okay, chiefly the ribs, the sternum, the pelvic bones, okay, the femur, those are the cells that keep on producing. Okay, so in the hematopoiesis, the original cells come from the multipotential hematopoietic stem cells. There are a lot of stimulators and inhibitors that exert their function here. So some of the stem cell will be directed to either become common myeloid progenitor or the common lymphoid progenitor. So this is the first interconnectedness between the two systems. So ini stem cell dia macam hub to control the hematology and the immunology system. So from the common myeloid progenitor, we get the thrombocytes at the end, erythrocyte, the mast cells, okay, mast cells, which can be also considered as part of the immune system, and then the myoblasts also produce a lot of the immune system cells, and then the common lymphoid progenitor, natural killer cells, and also the lymphocytes. Tadi tengok Shasha, I mentioned that the lymphocytes, their nucleus covers almost the whole of the cytoplasm. Kalau kita lukis pun macam ni lah kita expect people to draw. And then we have the plasma cells, the T lymphocyte, the plasma cell will produce antibodies at the end. Okay, any questions so far? Semua tutup kamera ni, tak nak tengok, tak nak apa, tak nak tunjuk muka ni. Malu semuanya.
Okey, doktor.
Okey.
Siap ke?
Setakat dah,
Ha?
doktor.
Okey Okay. So the organs. So we have the primary lymphoid organs and secondary lymphoid organs. Yeah, we have the thymus, bone marrow, then we have lymph nodes and spleen. The bone marrow is the place where the erythrocytes become mature before they are released into the bloodstream. Thymus, which cells will be brought to thymus and then mature there? Shakira. Shakira, are you there?
Yes, I'm here.
Yes, the T
T-cell,
cells.
doctor.
Okay. Okay, good. Right. I'm just checking whether you are still there or not. Okay. Changes in hematology readings. Okay. What can cause the changes? As I mentioned, firstly, age. Yeah. The babies will have different considered normal in comparison to the kids, and then to the adults, and then elderlies. Okay, sex. Okay, male and female, they have different reference range. Geographic location. Okay, geographic location ni satu tadi apa connect different in term of apa ni origin, I mean race, yeah, African versus Caucasian versus Asian. Secondly, people who are living in higher altitude will have a different reference range. Yeah, macam those Sherpa who are living in the Everest area, they have different normal range. Eskimos, they have different normal range than others. Okay, that the differences is due to the geographic location. Okay, so the pathologic changes in the values of the formed cellular elements occur either with disease or injury. When we talk about the normal values, apa sebenarnya normal values ni? Normal values ni is a result of huge studies across the regions of the world, and we calculate the normal values by calculating the mean for healthy individuals of the group. Okay, katalah kita ambil elderly, kita dah buat satu study elderly only. Apa maksud elderly? Apa maksud adult? Apa maksud normal ni tadi? Katalah untuk normal male and normal female. What we mean when you read the literature, bila dia kata normal values for men, normal values for women, yeah, it actually refers to those who are healthy and between the age of 25 to 40. That is in modern medicine. Kalau awak baca apa-apa normal range, normally itulah, kecuali if they state otherwise. If not, you can assume that then the mean is for individuals who are healthy, does not have disease, does not involve in apa ni, activities like smoking, etc., and age between 25 to 40. Okay, and we calculate up to plus minus 2 standard deviations. Okay, so when I think by this stage you have some insight of the importance of hematology and immunology service. Yeah, hematology, as I mentioned, is the first test that we normally do. I mean, in terms of the healthcare service lah, kita akan ambil blood and then look at the blood. We have some kind of idea what is problem, what's the problem with the person, or no problem with the person. But it is customary to take a blood sample and then interpret the blood results, and it is sort of It can serve as a screening procedure in case we miss something, or it can also serve as a diagnostic result where we can identify. It can also serve as a monitoring result. So katalah patient tu dah ada penyakit, kita treat, kita nak tengok kesan treatment, kesan intervention that we have given to the patient, whether it is improving or is it making things worse. Yeah, so a lot of things can be read, can be gleaned from the hematology results. Immunology results normally involve further investigations. Ya, kalau macam kita nak buat blood transfusion, okay, kita nak buat blood transfusion, we need to conduct immunology test first to see the blood groups, etc. So that one move from hematology to immunology test. So hematology service first. Hematology service includes firstly collection of samples. So they start from the phlebotomy, from taking the sample, and then blood cell count, hemoglobin assessment, and then blood grouping, and then transfusion medicine. Okay, to cover lah semua tu. Ada yang bahagian yang overlap dengan immunology, like the blood grouping. Immunology service pula include blood typing or blood grouping tadi, LG check, checking presence of autoantibodies, autoimmune diseases, as you have been taught last sem, actually covers a lot of diseases. Covers a lot of diseases. Okay, Eliza, for example, bukan Eliza, Eliza, and other types of tests, flow cytometer. It also involve in infectious disease serology. So this COVID-19, for example, masa ni the serology service is in high demand because when we want to detect for COVID-19, for example, we use a lot of serology as samples. We run a lot of serology tests to check for COVID-19 and also for development of new diagnostic kit and monitoring the patient. One thing that currently happening is that most of these procedures are now automated. So good news to you, maybe bad news as well. But good news is almost everything lah is automated these days. Okay, kita nak buat blood sample je yang tak automated ya. Maknanya you still have to go to the patient and get the consent to get the sample. Tu macam mana nak suruh machine buat consent semua tu? Boleh. Yeah. So you need to go to them and talk consent. You need to do the blood sampling yourself. Okay. The smear yang saya tunjuk tu, from there you can count. Okay. In the past we count every single cells. Okay. But these days you just put it in the analyzer, wait for 1 or 2 minutes, dah dapat result. INR for people with warfarin blood coagulation problem, we check for their INR level. So these days you just prick their fingers and then within a minute you get the count of the INR. Dulu kita kena buat manual, counting semua manual. Okay. And not only that they are automated, most of the machines are multi-analyte analyzers. Okay, multi-analyte analyzers. Macam tadi, you have the blood smear. In the past, you have to count how many is red blood cells, how many are non-red blood cells, how many are lymphocytes, etc. Okay, so in the first few years of the introduction of the automated machine, the machine could only count for red blood cells only. And then they introduced machine that could count the white blood cells only, and then the platelet only. Okay, but these days, the machine can count all different type of cells, from the red blood cells, white blood cells, as well as the platelets. Okay, Okay, Akilah, are you there?
Yes.
Okay.
Okay.
Saya boleh nampak Fahana. Fahana dari tadi dia istiqamah buka dia punya kamera. Shasha pun buka kamera. Anak saya yang kat sekolah, kalau dia orang tak buka kamera dalam kelas, markah dia tak dapat. Kalau dia ada pentaksiran sekolah kan, dapat sampai 6 ke apa ke. Cikgu dia takkan bagi dia 6. Sebab dia tak ada involvement dia kata dalam kelas. Bila saya tanya kenapa anak saya dapat 5 je ni cikgu, macam mana dapat 6? Dia kata, oh yang ni sebab sekarang ni COVID, dia assessment dia tak boleh nak buat macam dulu. So salah satu assessment yang dia tengok untuk dapat markah involvement semua tu, bila tanya soalan kena jawab. Kalau cikgu tak tanya soalan, student tu kena bertanya. So dia nampak ada proaktif lah sikit. Kemudian apa, buka kamera dalam kelas dia kata. Okey, saya harap kita tak payahlah sampai ke tahap tu pentaksiran dalam kelas di universiti pula. Oh, pening kepala ni. Kalau buat pentaksiran macam tu ramai yang tak dapat ni ni. Tolak 10 markah ni.
Switch
ada
on
yang
the
tengah
tadi buka doktor tapi sebab lagging, so saya tutup set.
Okay, alright. Ya, tak apalah, bukalah sekejap-sekejap tu tak apa. So that I can see that you are there. Okay. Dia kadang-kadang saya bercakap ni tak nak juga macam shock sendiri. Nak juga tengok student saya pay attention or not. Sekejap-sekejap kan. Macam Eva ni pun saya tak ingat mukanya walaupun buat FYP dengan saya. Siapa seorang lagi? Nadia ke? Entah ada entah tidak ni. Okay. So we have some human interaction lah. So masa COVID ni memang satu perkara yang bagi saya sangat challenging is having human to human interaction, not talking to the laptop. So tak payah buka all the time, but from time to time. Internet kat Mahalah memang lagging Nah, okey.
Kadang tak ada
okey.
Dah ramai.
Alright.
Kadang-kadang.
So you all need to go to Fahana's room. Pergi kat bilik dia. Okay. Study ramai-ramai. Cari tempat yang ada strong connection. Kalau bilik tak okey, macam saya, bilik saya, office saya tak okey. Saya selalunya buat dekat office di BMS. Kalau ni. Ataupun saya kadang-kadang saya juga lari ke library. Saya pun kena lari-lari juga. Or I'll be at my home lah. Okay. So where are we now? Okay, we have been talking about immunology, hematology, the blood cells, giving you some insight. Ini introduction je ni. Saya ni dah berapa minit dah saya bercakap ni. Dah pukul apa ni? Syahidah? Pukul apa dah? Tak dengar.
9.18 doktor.
9.18 tadi kita ni. Okay. Okay, I will start at 9:30 we take a break there. Okay. I will continue a little bit more. Okay. All right, this is an example of the a hospital floor plan. I could not find any floor plan for any Malaysian hospital. Yalah, dia mungkin confidential sikit. I could not find plan for SASMAC, for example. I found this floor plan from a hospital in the UK. So this is the kind of floor plan that you might be accustomed to in the next few years if you happen to work in a hospital setting. Yang ini hospital ni, awak tengok kat sebelah sini dia ada children's ward. Not all hospital has a dedicated wing for kids. Not all hospital has this. Only big hospitals. Ya. Kalau hospital yang biasa tu dia akan ada ward sajalah. Yang ini dia ada satu wing special. Some much bigger hospitals, they may have children hospital even. Children hospital. Kat UIA kita pun ada juga yang special. Kita ada dental hospital. You know, we have dental hospital at Kuliah of Dentistry, and it is the first dental hospital in Malaysia, if I'm not mistaken. But it is definitely the first dental hospital owned by a public university. I think by a university in Malaysia. No other university has a dental hospital. We also have, as a university, as a public university, the first pharmaceutical plant. Maknanya kilang membuat ubat. Universiti lain tak ada. Do you know that?
Baru tahu, doktor.
Nama dia ICOP, ya, ICOP Sendirian Berhad. It is part the plant, the factory, ya. Factory plant maksudnya pilot plant maksudnya factory yang kecil, ya. Dia kecil. Dia masih lagi kecil tapi dah start producing. If you go to our labs, you might be using iHand. Awak tengok botol hand sanitizer tu, nama dia iHand, ya. And kalau awak baca dia punya manufacturer dia is ICOP Sendirian Berhad. ICOP Sendirian Berhad is owned by UIA, and they have their pilot plant, pharmaceutical plant within Kuliah of Pharmacy. Okay, so kalau awak nak buat internship, you can also apply to ICOP. Apply to ICOP. You can learn on how to manage the management part, on how to manage pharmaceutical plant, or you want to get involved in the pharmaceutical process. Macam mana nak buat ubat? Proses-proses yang terlibat. I take sometimes saya ambil FYP student. This year pun actually I offer extra two topics, ya, related to pharmaceuticals, tapi tak ada orang ambil. Okay, anyway, coming back to this floor plan, ya. So forget about the children part, but if you look at the main hospital here, okay, so these are you can see the labs, ya, pathology labs, medical physics, angiography, cardiac suites, coronary care, speech therapy. Here you are, blood test, and then urology of patient. So as biomedical graduates, you could actually be at any of the labs. Even the speech therapy pun sebenarnya awak boleh masuk kalau awak nak. You may need to take some special courses lah before you get into ni. Sekejap saya on saya punya charger. Alright, but here you can see the blood test or the phlebotomy or the hematology lab, yeah, is central. It's like the heart of the hospital. Normally kita akan letak the blood collection center ni in the most convenient place so that it is like in the middle of the hospital. From everywhere, people can have a equidistant walk towards it. You know, ward kat belah sini, ward kat sini is around the phlebotomy. So this is one of the good floor plan lah. You will come across hospital which have a horrendous design. Okay, so nanti blood test dekat ujung sana. So everyone need to walk very far. Okay, to get there. There are now hospitals, very big hospitals. For example, they have system where from the ward you can put it into the vacuum system on the wall, masuk dalam tu, dalam kelalang dalam botol dia tu, nanti dia akan sedut terus hantar ke lab. Ada juga yang macam tu, ya. So urology. So when you get into the labs, okay, for hematology and immunology, sometimes you will not find any hematology lab. You will not find any immunology lab because it is combined with pathology labs in some hospital. In another hospital, it may be part of biochemistry lab or biochemical lab. But in some hospital, they may be independent unit, hematology lab, immunology lab. Sometimes it is immunology and something, and immunology and serology. So this arrangement depends on the size of the hospitals. Small hospital tend to combine, ya macam saya tengok dekat Muazzam, Hospital Muazzam Shah tu, dia satu area yang besar. Kemudian ada section dalam tu. So dia ada hematology dia combine dengan yang lain dalam satu area. They only have section and they share their equipment. The only place that is different from that lab is the blood bank, which is in a different room. And actually, despite the name, blood bank tu macam nampak macam besar kan. Dekat Muazzam Shah dia satu fridge saja. That they call that blood bank. It is managed by the lab people. The same official yang manage lab is the same as the one that manage the blood bank. Normally dia macam tu lah. There's one main supervisor controlling the lab and the blood bank, and then the supervisor sendiri. In the UK, the blood bank must hold a biomedical degree. Dia tak boleh degree lain. Kalau dekat UK lah, must hold a biomedical degree. In Malaysia, because biomedical science has just been introduced, macam dalam 10 years ago, 10 years ke, saya dah masuk 2006, so dah more than 15 years. Masa saya bawa balik dulu, many of these labs supervisors, blood bank directors, they are non-biomedical. They come from biology degree holder, biotechnology. They took some courses and then they fill up the position. Okay. So laboratory medicine service, that will be the parent name for many labs. Ada blood bank, hematology, sometimes it is on its own, sometimes it is part of biochem lab, sometimes it is part or combined with the immunology, sometimes it is part of the histopathology. It depends on the arrangement in the hospital. So these are the examples of lab that you may be involved with in the hospital clinical setting. Okay, right. So we stop here. Yeah, for how many minutes? Mana Shakira?
5 minutes.
5 minutes. I give you 10 minutes.
Okay.
Boleh?
Boleh.
10 minutes. All right. Okay, Assalamualaikum. Okay, saya ada bagi attendance link. Can you take some time to just click to record your attendance? Okay, Nadia Kamaluddin. What can you see on the screen?
Slide Hematology Lab test.
test. Okay, have you opened the attendance link? The attendance link has been given in the chat box. Anyone who has not clicked the attendance, can you take some time to do it? Yeah. Otherwise, the list of the tasks. For some reason, you cannot do it now. You can have until Sunday to do it. All right. So just before the break, we were talking about the laboratory medicine services and the hospital floor plan. Okay, so let's go and look at what kind of common lab tests there are. Okay, so this hematology lab test may include RBC count, very common. Everyone have experienced this. Yeah, nak masuk UIA kena buat semua ni. Yeah, full blood count, full blood test. Yeah, so in the full blood test FBC, kat Malaysia dia panggil apa kalau UK dia akan sebut FBC lah, full blood test or full blood count. So when we say full blood count, it means the 3 main elements, the red blood cell, white blood cell, and the platelet counts would be tested. And then some other parameters these days are also included, like hematocrit, mean corpuscular volume, mean corpuscular hemoglobin concentration, MCV, MCHC. In the past, we need to get the first three first, the values for the first three, and then calculate manually using formula to get the hematocrit, MCV, MCHC, PCV, etc. But these days, because of the automated machine, all of this will be given by the machine. Yeah, you just need to press print button. Dalam masa beberapa minit je dah selesai. Okay. Now, these are the common lab tests. Under normal conditions, the production highly regulated process, quantitative and qualitative hematology abnormalities, yang semua ni, all the production, release and survival will be reflected by the values that we get here. Okay, so they give quantitative and qualitative values. The abnormalities may result when they release and survival. Okay, so our body system is very, very good actually. Yeah, when we have slight deviations, most of the time you will not even feel the problem. Okay, macam sini ada berapa ramai student perempuan ni. Like more than 95% of you here are all ladies. Yeah, I do not know how many of you may experience longish period menstruation. How many of you have or common normal for you to have heavy menstruation? So during the menstruation, if it is prolonged or heavy, you may actually experience anemia. Okay, anemia. So maybe kalau mengantuk dalam kelas tu mungkin sebab dia anemia. Tidur je kan, because of the tiredness, because of the anemia. Dia malas nak kerja bas, malas nak datang lab lecture. It could be because of that, but in term of quality of life, bila nak makan bersemangat pula pergi nak pergi kantin kan. So dia anemic, but the anemia does not affect most of the activities lah, can still walk, run, because the body system is very good. These slight deviations can be masked or can be corrected, by the body. How do they do this? By increasing cell production, for example. during menstruation, when there are a lot of blood being lost, the body will be increased in terms of the cell production. Okay. Tapi apa tak boleh lama lah. So during menstruation tu mungkin kalau you are the heavy type, you may want to consider adding into your meals things that can help the body system, ya, macam benda-benda yang boleh meningkatkan yang ada banyak iron, ya, iron rich food, so that dia boleh tolonglah body walaupun tak rasa macam okey je, tapi it's only when we do the lab test we can get a quantitative assessment of the anemia level. Otherwise, the body appears to be normal. And in fact, many of chronic diseases, okay, when they are in the early stage, they are not detected because the patient feels okay. Okay, dia tak datang pun ke hospital because dia feels okay, can still do normal routine. Okay, only when they are totally unable to move, etc., then they are being referred to the hospital. And then when we do the lab test, kita tengok, oh, actually dia dah jadi chronic. Okay, so this is the normal range that you need to commit to memory. So I may ask in the OSPI to write down what would be the normal RBC level for a male adult. So kena bagilah. Note the units. Okay, full mark will be given if you give correct unit. Okay, correct range. Okay, I appreciate that in different countries and in different textbooks, even on different websites, okay, slight variation occur. Macam contohnya kita tengok RBC ni, you may find a reference textbook giving 4 to 5 only. Okay, slight variation, but it's more or less the same. Because apa tadi? Ingat saya cakap tadi plus minus 2 standard deviation, so we allow for different systems to have slightly different, but dia akan hovers around the same value. Okay, for the purpose of standardization, this is the one that I will ask you to commit to memory lah. Nanti bila bekerja you kena tengok lab tu pakai reference range mana, apa, ya. This is true for all type of procedures, ya, all type of procedures, because procedures ada banyak sebenarnya. And it depends on that particular lab. Yeah, I will share my experience. Okay, masa saya buat masters, prior to doing my masters, saya dah biasa dah buat electrophoresis. Electrophoresis ni kita kena create satu gel, and then we run electricity so that our material tu akan separate according to dia punya density, size. Yeah, so I've done it before, and when I did my masters, I went to a different university than my degree, and I was one of the things that I need to perform on the first day was electrophoresis. So the new lab gave me a protocol for electrophoresis gel making. Tapi sebab saya pernah buat sebelum ni, I just do it according to the one that I knew before. And I could not make the gel. Saya tak boleh buat gel tu. Gel saya tak jadi. Berkali-kali saya ulang. Until at the end of the day, I went back to my supervisor and told him that I have problem making the gel. I do not know why. And he simply asked, did you follow my protocol? Of course saya tak follow. Bila saya tengok, oh, dia ada slight variation. Dia ada slight variation. And it could be due to the electricity of the place. Ya, kat universiti baru tu dia different sikit. And maybe also the gel maker tu ada sedikit different. Also the material the company that produce the material, the SDS solution, for example, they're different sikit. So the learning point is when you move from one lab to another, you need to check what is the new labs protocol and follow what work there. Apa yang boleh dipakai di tempat lain to follow the protocol, okay, of the place. Okay. Right. So before we get to that lab value study, we need first to get our sample. Yeah. So hematology and immunology memang kena ambil blood. Yeah. It's not like pathology. Pathology dia mungkin pakai urine, dia pakai stool. Okay. In hematology and immunology, main thing is blood sampling. Yeah. So there are a few ways to get our blood sample. One of the most common and everyone has experienced this is by taking it in this way. Apa, ambil dekat tangan. Okay. Siapa yang belum pernah? Tak pernah. Tak pernah. Takkan tak? Eva tak pernah ambil dekat ni. Phlebotomy ni. Sure. Kenapa tak dapat? Kenapa tak ambil? Bukan saya kata awak yang ambil. Orang lain ambil pada awak.
Oh, kalau demam darah tu ambil lah kan?
Ambil lah tu. Mungkin awak tak pernah mengambil daripada orang lain. You have not taken from others, but blood has been taken from you using this method, kan? Meaning that you look here, ada tourniquet, ya, warna biru ni tourniquet. So it compress your vein, and then somebody is using the picagari ni to take out blood. Yeah, so this is one ni. Tapi this guy dia letak atas kerusi. Actually not that good lah. Yeah, in this is emergency kot, so dia letak macam ni. And then the hand is hanging. Okay, is this a good practice to do? Is this a good practice? Ada orang ambil tangan awak dalam keadaan macam ni tak? Okay, you look at this guy. Okay, this guy appears professional. Dia pakai baju dia, okay, professional. So dia juga pakai glove. Okay, very good. Okay, angle of the syringe is good. 30 degrees angle, 45 degrees angle. Okay, good. Patient has been put on, tourniquet has been put on the patient. The problem is here. Okay. So, kalau kita tak tahu what to do, what is the right practice, okay, when we first look at this picture, it looks okay, okay, but once we know what is good practice, we can evaluate, okay. Macam kita tengok drama pun, kadang kita tengok drama, orang tengok macam okay je, oh, tapi bila kita tahu, banyak benda kita boleh kritik on the drama. Okay, this is one of the ways to get blood sample from the arm. This is another common method to get blood sample, especially if we do not require to do a lot of tests. Okay, contoh macam awak nak test satu parameter je, INR sahaja, then we can do finger prick and then put it and test quickly. Yeah, the previous one, blood sampling through the vein or through the artery, that is performed when we want huge volume of blood, perhaps because we want to run a lot of tests. So kita kena ambil banyak. Also, we do the finger prick when or capillary, kita panggil capillary sampling, when we cannot do the phlebotomy, we cannot take blood from the vein. Contoh macam mana? Contohnya orang tu burn victim, terbakar, banyak kawasan body dia terbakar. So macam mana kita nak ambil? So we can do capillary sampling. from the fingers or any other areas that are suitable. There a lot of areas sebenarnya that we can take our sample from. Yeah, common here lah. Okay, look at this. Okay, dia ambil dia buat finger prick dia dekat mana? This is perhaps the right hand, perhaps the jari telunjuk kat sini jari telunjuk. But look at the area on that finger. The prick was not done on the panggil apa ni, perut jari tu. Okay, this is the practice. We never do the prick here. Why? Because here satu dia sangat sensitive to touch. So when we do the prick, it could be much painful. Satu. Okay, so we do it at the side. Secondly, this area is for identification. So in case kita buat prick kat tengah ni, dia kacau jalur ni. Dia kacau jalur ni, nanti orang tu akan bermasalah for any identification. Although this is not done on the thumb, dia tak buat thumb, tapi any rule of thumb, kalau thumb tu bermasalah, contoh thumb tu cedera, okay, cedera, terputus contohnya. Dan for identification purposes, we want to use other fingers. So we avoid lah this area. This area ni yang tengah ni awak nampak ada bulat kat sini. This is the area that is specific for every person. So we do on that side. So when we say blood sampling, it's not just something which is to be taken for granted. There's a lot of other considerations to be think of. Okay, blood sampling can also be done on the earlobe. Ni earlobe. Ni orang ni. Okay, there are some tests that we can do or it is considered good to do sampling on the earlobe. Here, A ni area where the puncture can be done on. Kita boleh buat dari atas sampai ke bawah. Kenapa kita tak buat kat sini? Sebab kat sini dia ada tulang rawan. So kalau kita cucuk dia lagi takut damage the tulang etc. and the wound healing would be less it would be slower lah than to do it here. Here the a lot of capillaries as well, so we can get good or enough amount of blood, and then easy to heal. Easy also, convenient also for us to do the prick. So but the most common kat sini lah, dekat kita punya earlobe. Kat situ ada area yang banyak sikit lemak kan. Okay, some people do not have this earlobe, dia macam straight je dia punya telinga. Tapi tak apa, boleh je buat kat area yang macam orang ni. Orang ni tak ada lobe sangat. Okay, now then we use the lancet. Okay, lancet kita boleh pakai apa-apa jarum, but these days we use lancet because lancet ni kita dia dah specifically design so that when we press dia hanya masuk beberapa milimeter ke dalam, enough to get the blood. Kalau kita buat sendiri manually, we can still get tapi kita takut kita tercucuk lebih dalam. Okay, so it is also good to have this commercially available lancet. So we get the blood sample there, we press, and then we can collect. We can use pipette, okay, or we can use directly collect into what we call micro cuvette. Okay, micro vial. Letak je kat situ, darah tu kena kat situ, dia masuk ke dalam. Okay, so this is not commonly done lah. Commonly we do the venesection, also called the phlebotomy. So this is micro cuvette. Dia ada macam-macam shape. So ada yang macam ni, ada yang macam ni. The point is to get the blood sample into the cuvette, and we can keep and carry easily for further processes. Ini yang biasa awak tengok kan, the collection tubes. Okay, perhaps you have not been introduced to other type of blood tubes. Maybe the one that you have commonly encountered is this number 5, EDTA tubes. It has a purple or pinkish color. cap. Ya, ini yang biasalah. Ya. Selalunya orang masuk hospital kalau kita tak ada nak test yang lain-lain, only full blood count, the screening test, they will be we will take their blood and use the EDTA tube. Ya, tapi there are cases in which we want to do other type of tests, so we will use also other collection tubes like this. Ya, ini glycolytic inhibitor ada dalam ni. This is specifically for people with diabetes. Ya, kita nak tengok dia punya glucose level. We put the blood sample into the glycolytic inhibitor, the grey cap tubes, and then that one will be adalah dia punya proses dia sendiri. Kemudian kalau tengok kat sini ni ada blood culture. Ini kalau kita suspect that the person has infection in the blood, kita nak culture the microbes. So the microbe kita tak letak dalam vial-vial yang lain ni, kita letak dalam specific bottle ni, blood culture tube or blood culture bottle or blood culture vial. There are one for the anaerobic, one for aerobic. Kena baca label properly. Okay, okay. And then when we do the collection tube, when we do the collection, if we want to test for many sample, katalah Shasha, kita nak tengok sama ada dia ada blood infection, dia punya glucose level macam mana, blood count dia macam mana, macam-macam lah kita nak test kan. So we will be using a lot of blood. So dari tadi, dari satu sampling tadi tu kita akan masuk ikut turutan. We cannot put the blood sample into the glycolytic inhibitor tube first. This one is last. The anaerobic aerobic vial ni dia kena first. Sebab kalau kita masuk dalam tube ni dulu, kita takut kod-kod dekat sini ni, dekat atas ni dah ada dah bacteria, and then we will be transferring bacteria from the cap to the vial. So it will disturb our test. Okay, dia tak reflect what is actually in the blood sample. So the blood culture is the first to be filled up. So this is the sequence, yeah, sequence. So look, you guys are upgrading your knowledge, yeah, in terms of blood sampling, the different type of sampling, the different type of collecting methods, yeah, the different type of tubes. So it's not something to be taken for granted. Okay, from the blood sampling, we'll do blood smear. Okay, we do blood smear. The aim of the blood smear is to create one layer of cell. Macam ni kan, one layer of cell, kita nampak sebiji sel ni tidak bertindan. They are not overlapping. So we can see clearly their lobes. Ini ada 4 lobes. What do you think they are? What do you think they are? Okay, this one is big cell, big nucleus, yang almost completely filling the cytoplasm. Shasha mesti dah tahu dah ni, sebab dia dah kena tanya tadi. Lepas ni Shasha memang akan betul lah yang ni kalau soalan ni. Tengok ininya dia dah tahu dah. Okay. So we want the smear to be one layer cell. Okay. Tengok ni dia ada shape-nya macam ni, macam feather, macam bulu burung kan. Okay, in the lab, you will be dropping one drop of blood here, tapi kita nak you create this kind of smear. Okay, smear yang kat sini dia tebal, kemudian dia menjadi nipis, kemudian dia membentuk feather shape. Okay. Feather shape, and then when we look under the microscope, they are all single layer cells. This is a good blood smear, and it takes practice. We'll be giving you one box, fresh box. Pakailah, so jangan takut sekali buat tak jadi. Saya boleh guarantee lah, first time you do it, 99.99% of people will not get it right. Okay, kadang-kadang dia drop blood banyak, sometimes dia tolak salah. Nanti kita tengok ada yang zigzag sebab masa dia nak buat tu dia terketar-ketar tangan dia. Okay, So sekali dah buat, nanti dia akan confident, dia akan jadi okay. Okay, so you need to do many times. Student normally will use up all the fresh slide. Don't worry. Okay, pakailah habis-habis, tapi satu kotak je lah, satu group. Okay, satu kotak, satu group. And then after you have, because we are just practicing. So lepas dah habis, katalah dah tak ada, dah satu kotak dah habis, you can wipe and then you can clean and then you can reuse the slide. Yeah, reuse the slide. As long as they are clean, they have been dried, then you can practice doing the smear. Okay, so don't worry. Yeah, I want you all to try. Okay. These are the variation. Bila kita tengok dalam bawah mikroskop ni, this is normal. These are all normal. You can see the red blood cell, the central area of pallor, etc. But in the hospital, you see many other types of variants of the red blood cell. Ni red blood cell only. Yang white blood cell ada variant juga. So this is the normal one, and then the microcyte, and the macro. So if you do not know what is normal, you will not be able to differentiate the normal with the abnormal. Nampak macam sama je, tapi bila you dah biasa tengok, you akan tahu, oh, this is actually bigger. And then when we do a test, we run the test in the machine. The machine will be able to calculate the diameter and give us a result which is more definitive. Not subjective. Subjective ni maknanya awak tengok, awak rasa-rasa je dia besar. Bila kita letak dalam mesin tu, memang bacaan dia, diameter dia besar. So conclusive lah that it is a microcyte. Oh, bacaan dia kecil, so it is a microcyte. Okay. But other shape like oval microcyte, this cannot be read by the machine. Macam mana mesin tu nak baca oval microcyte? So looking under the microscope, look at the blood smear slide. Tak boleh tidak, kena buat juga, because macam ni acanthocyte, sickle cell, still tak ada mesin yang boleh baca lah setakat ni. We can get some clues, but at the end we still need our two eyes to then look and decide. Okay. So ada red blood cell inclusions. These are all cannot be seen under, cannot be detected by the machine that we have. They need to be seen by our own eyes. Okay. Different erythrocyte variation. These are normal for erythrocyte. Ini yang saya tunjuk ni yang abnormal because of the disease state. But this variation can also be normal. Ni contoh because of the development Kenapa saya letak erythrocyte? This should be neutrophil variation. Nanti saya tukar balik. So as the neutrophils mature from myeloblast to the mature neutrophils, they start to produce granules. So you see here some granules. In the beginning no granules, but here lot of granules. Tapi granules dia tak macam eosinophils. Granules dia nampak dekat luar, tak cover the nucleus. Okay, tapi dia ada multi lobes, 3 and more. Yang ni band cell is the immature neutrophil. Jangan ingat band cell is not neutrophil, it's still neutrophil, but band cell. So how do we differentiate all this? It comes with experience. First, we understand that there would be variations of the cells. Some variations are normal, some variations are abnormal. Okay, the one that are normal ni lagi tricky nak tahu, so that we don't make wrong identification. But it requires experience. Experience. Okay, you can read later on lah why there are different presentation of the neutrophils. So this one become more advanced knowledge. Okay, ni hypersegmented neutrophils. So tadi saya dah cakap neutrophils is a cell which has granules. Okay, you can see a lot of granules kat luar ni. Okay, granules here, granules ni semua granules ni. Okay, and then dia ada multi-lobe. As the neutrophil develop, nucleus dia akan become lobulated, dia akan lobe. Tapi dia bila dia dah tua ataupun dalam certain disease state, the lobe is separated. So kita panggil dia hypersegmented neutrophils. So these are all abnormalities. So these are some of the example lah. So you can see the lobe. Some of the lobe appears like it is totally separated from other lobes. Very thin layer of bridge. So saya dah tunjuk how we take blood sample and then we do smear, we look under the microscope, some of the variation of the cells. We talk on all those things later on in individual lectures. So some of these sample of the blood cells, we also run for the immunology test. Banyak immunology test that we can do. We can do for blood grouping, for example. We can test for antibodies, for example. And the purpose of the test will dictate the different type of tests that we will do. Okay. Contohnya kalau nak buat blood grouping, kita buat Cum test. Cum pula ada beberapa jenis, ada direct and indirect Cum test. Okay. And then for antibody macam ni, we can use Eliza. Okay, Eliza. So here on the screen, you can see Eliza ni dia pakai microplate. Small amount of sample are being tested using this plate. This plate awak dah pernah jumpa kot dekat dalam microbe pun kita pakai. Very useful thing, very useful thing. Genetik pun kita boleh pakai microplate ni, versatile basically, dan kita ada satu benda yang kita panggil microplate reader, small machine but very useful, very versatile. You can run many different type of tests on it. You just need to change maybe the frequency, the light frequency to read, and then you get different colors, etc. when you put in different substrate. So the basic theory ataupun the basic concept is that on the there are different type of receptors. Okay, macam antibody ni pun dia macam receptor lah juga, on the surface or in the serum. That are specific, kalau macam in term of infection, specific antibody will bind to specific receptor of the invading pathogen. So kita pakai that concept, then we use the antibody that we have tagged it with a certain type of enzyme. Sometimes this has a fluorescent dye. Okay. Then we use the antibody to capture the antigen that we want to detect, and then using the dye we can then identify the strength of the amount of the antigen, etc. So this has been used for COVID-19, like the SARS-CoV-2 SRBD kit. We use the concept, we use the ELISA to detect the spike protein receptor binding domain of the COVID-19. So dia dekat sini, kita pakai antibody that will bind, and then this one is for the antibody. Dan kita boleh pakai satu lagi antibody, anti-human IgG, for example, that we have label here, HRP or the horseradish peroxidase. We can use many types. Ini bukan satu-satunya, many types. And then we can identify lah, maybe a change of color, maybe the machine will shine at different frequency and then detect different type of light being emitted. Itulah dia punya konsep. Ya. So these are all using the immunology, the knowledge of immunology to be developed into diagnostic kit. So dalam kelas dalam lab kita belajar ELISA, but we cannot offer the ELISA test lah. It's expensive, ya. It is expensive for our purpose dalam lab yang kita nak buat. Dia tak sesuai lah. So kita akan buat blood grouping. I hope that at least you can do one immunology punya test, blood grouping at least, because that one is simple. We just need blood sample and then a simple kit. The concept is basically more or less the same. Dia jadi mahal ni bila kita nak pakai antibody ni. Kita nak labels pun dia jadi mahal because of that, because the process to create them is laborious. So dia jadi mahal lah bila kita nak beli daripada orang. Okay. I've been mentioning about automated instrument. This is one of the multi-analyte analyzer for blood sample. You see, they are holding the purple cap. Purple cap tadi apa? EDTA tube. EDTA tube sebab dalam ni ada EDTA. Ya, dia prevent the blood from clotting. So lepas kita ambil daripada orang tu, kita boleh bawa ke mana-mana dulu, and then reach our lab, and then just put this tube into the blood sample. Dia akan sedut masuk dalam, dan dia akan proseslah dalam ni. So all at your fingertip. You just need to learn where to press. Okay. So in terms of training, not much training needed. But the machine, one machine is cost us a bomb. Okay, costly. This is the Eliza microplate reader that I mentioned. So you put the microplate reader here, you put in. Tengok dah ada apa, button dia 3 je. Start, stop, lepas tu apa ni, pause. Kat sinilah ada button sikit where you can change some parameters. Okay, according to the sample type and according to the type of test that you want to carry out. Sikit je. So nothing to be afraid of, nothing to be afraid of. And when you get into your internship, you will come across all this machine. It's not difficult to handle. Right. So that is one kind of situation, one kind of setting, where you are in the lab, in the hospital setting. You take blood sample, you do run a lot of tests. Some tests are manual. Macam blood smear tu memang kena buat manual. They are developing machine, automated machine to do blood smear sekarang ni. Ada. Tapi tak pakai banyak lagi lah dekat satu dunia. Sebab dia tak berapa, ada banyaklah benda yang ada beberapa bacaan-bacaan yang masih kena baca dengan 2 biji mata kita. Tak ada yang boleh dibaca dengan mesin. So teknologi tu dah mula ada lah, nak katanya. So maybe one of you here, although you are in a diagnostic pathway, you may want to develop something. Okay. Kalau awak, apa nama dia, kalau awak aware, there is one issue sekarang ni with, saya lupa nama benda tu. Tapi this lady has been trying to develop a way to do the blood testing using laser or some kind of light technology. Awak tak perlu lagi ambil darah macam tu. Dia dekat ni je. Saya dah tengok benda ni 10 years ago in USM. They try to do this, just by shining some laser onto your blood vein, and then kat situ kita buat bacaan RBC, buat blood cells semua. So no invasive procedure. Akilah macam berkerut dahi, It sounds like too good to be true. Macam dia percaya ke ni, But 20 years ago, I was discussing with my English teacher on the Malaysian ambition to create a Silicon Valley. Now we call MSC. We call Cyberjaya tu lah kan. And then at that time, we were talking about how in the future we will have smart house where we can lock our door using handphone. We can cook our rice. We can cook we can turn on aircon from our office. So katalah lagi 10 minit nak sampai kat rumah ni. So nak masuk rumah ni aku nak sejuk terus. Tak nak tunggu aircon ni. So sekarang dah aku nak onkan aircon tu. Contohnya. So my English teacher at that time dia kata, it sounds too good to be true. Boleh ke kita buat macam tu, dia kata. That was 20 years ago. But today, yes, you can do that already. We can even have a CCTV, boleh nampak CCTV boleh control from our fingertips je. Lupa nak buka CCTV masa balik keluar rumah tadi, boleh test. Kalau ada orang pencuri nak masuk, berbunyi, dekat handphone kita berbunyi juga. Kita boleh tengok CCTV yang mana satu, kat mana dia nak masuk tu. Apa lagi? 20 years ago, we cannot do this. Okay, apa dia panggil? Zoom ni. We cannot. Kita tak boleh buat. But today we can. Now ada breakout room lah, apalah lagi kan. Macam-macam. Apa nak cakap tadi? Okay, I was talking about the technology lah. USM. Yang tu dia nak buat tengok tanpa membuat blood sampling. Hanya melalui laser. So maybe one of you want to develop that idea further. It will be perhaps very good to do. Okay, so there was itulah yang pasal lab setting in the hospital, etc. Another service that is closely related to hematology and immunology is the blood bank. Blood bank. This is a setting in the blood bank. Contoh lah. So you got the patient, the personnel. Maybe one of you will be this personnel later on. Someone is waiting, so dia ada tempat dia wait. This is a very good and nampak macam apa kata well-funded blood bank lah. In reality, not many can afford this kind of setting. Yang awak pernah tengok mungkin dekat mana? Pasar raya. In the mall, kat tengah-tengah tu orang buat blood donation. Kan? So ada patient dengan personnel, bag dia tergantung ke tepi ni. Tak adalah mesin-mesin semua ni. Tapi ini yang perfect ideal situation lah, where people can come, they have a place to rest. Another person here, and then this is the machine monitor the patient, monitor blood, heart rate, semua sekali lah. So these are all the blood is being put in this machine. Dia boleh detect macam-macam dah kat sini. So the patient not only donate blood but also get some blood sample tests also here. And then here you can see some blood products lah. Some blood products, different type of blood product here. Maybe this is whole blood. This is tak tahu apa benda. Another ni kata blood processor, blood processor. So then you get the red cell unit. You can get some other blood products. And then the facility is also ada storage, ada transporter. Okay. So in certain hospitals like in Kuantan, for example, kita ada ramai orang yang donate blood. We are almost never run out of blood supply. But imagine rural area, macam Raub, contoh Raub. Okay, dia punya blood bank dia very small. As I mentioned, Muazzam tadi pun dia ada satu fridge yang kecil. Berapa banyak je dia boleh simpan. So kalau ada accident, satu kereta accident, takkan accident satu kereta, mungkin yalah kereta mungkin terbabas. Okay. Kalau accident dua kereta, ada 10 orang katakan. 10 orang darah lain-lain group. Okay. Sekejap dia dah habis. Maybe tak cukup pun. So they have to call people in Kuantan to send the blood to blood supply. So how do we carry the blood to Muazzam? How do we carry the blood to Raub? So you need a personnel well trained to do that. We need transport mechanism, van or buses that are air conditioned to transport the sample. So that is part of the blood bank service. Okay. So these are some of the apa ni panggil blood product. So we get the donated blood, and then some will be filtered to remove leukocytes. We get red cells, pooled platelet, fresh frozen plasma, very important. Okay, ini yang saya tunjuk ni from UK lah. So UK dia kalau katalah dia dapat source from non-UK source. Contohnya Farhanah melanjutkan pelajaran buat masters dekat UK, insyaAllah. And then you want to donate blood in the UK, so you are non-UK source. So non-UK source mean in your blood you may be carrying some kind of materials, mungkin infection ke apa ke yang UK tak nak lah, so they will do some other test before yours can be used for patient. Masa zaman saya dulu dia ada panggil Creutzfeldt-Jakob disease, at that time was very prevalent. So bila saya balik Malaysia, I could not donate blood because I have spent more than a year in the UK during that time. Okay, so I cannot donate blood because of the risk of transmitting Creutzfeldt-Jakob disease. So only in recent years Malaysia dia allow people to that have stayed in Europe during that time to donate blood. Okay, so the central blood bank and the hospital, the ward, the labs, as well as remote point of care. Contohnya ada outpatient clinic elsewhere that are linked to the hospital. They will be linked together by a lab management system, lab information service contohnya, central database. Semua kita sekarang ni dah centralized, kita semua key in dalam sistem, masuk dalam satu database that then we can use for our clinical management system, and finally to the patient. So this is another thing that is very important for you because many of you will become lab supervisors, blood bank directors. You need to ensure that there is this lab information service also. So kita cakap pasal Hematology Lab, Immunology Lab, itu lab. Tapi data yang kita dapat, macam mana kita nak selaraskan? How do we want to coordinate? So you need the IT people as part of your team. You need to make sure that it is good, it is usable, at the end can be tailored to the patient need. Okay, itu semua biomed punya graduate lah punya kerja. And you can, even if you like to do IT stuff, you can be the developer of the system. Okay, this is the blood bank. As I mentioned, the blood bank does not have to be very big, huge area. It could be as small as a fridge. This is a fridge. Tengok ada A, B, O, A, B. This is how we commonly arrange our blood sample. A type is at the top, B, O, A, B at the bottom. Kenapa dia tak ada apa-apa lah, dia setakat standardized ni. So kalau awak move from one hospital to another, you know, dia tak adalah tunggang terbalik kat hospital ni A, B atas, nanti awak tersalah ambil. Okay, so there are parts of the fridge that you need to know, and you need to maintain, semua ni. It's not a simple run-of-the-mill fridge. There are certain SOP that we need to adhere to, especially for the temperature recording. Because there is a high risk of infection being transmitted through blood. Kalau at certain temperature, some of these invading pathogens, bacteria, for example, they can replicate if they are at a higher temperature. So we always want them to be in a low temperature. And there are separation between the different types so that we don't make mistake. Okay. Apa lagi? Okay. Right. Any questions so far?
No.
Syakira dah pukul berapa ni?
10.35.
10.35. Okay. Hopefully I will be faster after this. Yeah. So steps in the investigation of a patient. Firstly, patient history. Some or most of the time, people who are working in the lab, they don't have to take patient history, but you still need to know some history. So sometimes dia tulis dekat note, ya. Sometimes dia tak tulis patient history. And then physical examination, normally lab people do not have to conduct physical examination. This is in the doctors lah. Cuma dalam case where you want to take blood sample, for example, you may want to do some small physical examination. Macam bila kita ambil darah daripada tangan, you need to know whether the patient has had, katalah dia perempuan, mastectomy, okay, to the breast. So katalah dia ada mastectomy on the right side of the breast, on the right breast, yeah. So we do not take any blood sample from the right hand. Okay, on the right hand. Atau dia ada limb adenopathy dekat bawah ketiak belah kanan. So we do not use that right hand to take blood sample. So some patient history sometimes is needed in that cases. So physical examination, kita tengok dekat tempat we want to take the blood sample. Is there any scar? Is there any wound? Itulah. So just very little physical examination. And then we send the sample to the lab, the relevant lab to do the lab test. They may need to be further investigated using imaging techniques, X-ray or CT scan, MRI, okay, and then diagnose. We get the final diagnosis, and then be treated, and then evaluated. Evaluation is important sebab sometimes kita misdiagnose, sometimes not partially correct diagnosis. Okay, so we evaluate, and when we evaluate, we need to do the flow again. Okay, this is where most of the biomedical graduate will be involved with, yeah, hematology, pathology, urinalysis, microbiology, and blood bank. Okay, this is the lab request form. Most of you will become accustomed to this. You will get this every day in huge number. You are what we call the gatekeeper to the lab. Yeah, when you receive the sample, you need to analyze the lab request form. Okay, ini contoh ni saya ambil daripada UK, but it will be the same in Malaysia as well. Ya, ni saya just ambil daripada internet. So in the UK, they have NHS number or the patient number. Okay, sometimes you have the hospital numbers. Not all hospital have this. What is important is the date of birth and the name of the patient. Okay, address ni tak penting. Specimen collected is important. Specimen that has collected maybe more than 24 hours ago is considered expired. You need to take a fresh sample. Signature of the medical officer is very important. The medical officer or the one who conduct the blood sampling lah, because they will be they have to take responsibility of the sampling. And also if we want to reject, we will call them to do the blood sampling. So they need to take, yeah, they need to take what sample, what test to be done, and those at the lab will have to check whether the tube is correct. Yeah, kalau dia tick kat sini full blood count, patutnya ADTA level the top tube, tapi dalam form ni selalu come together with the small bag, yeah, containing the tube. So if the tube is not there, so you need to call the officer involved and chase for the sample again. So these are things that you will be the gatekeeper at the lab before you run the test. Yeah, you will be deciding whether you want to proceed with the lab test or not. And then katalah dia ada lavender top tube ni, EDTA tube ni dia ada, dia dah tick okay, semua maklumat dah ada semua. You need to check the tube whether it is leaking or not. Contohnya, you need to check the sample. Is the sample already clot? Dia tak boleh run. Okay. So those at the lab is playing a very important role in the management of patient. So hematological practice, immunological practice, laboratory, clinical laboratory is very important, very honorable practice. It has both the clinical and laboratory components and require each other for successful completion of patient care. Yeah, this is the flow, specimen collection, and then will be sent for the specimen reception at the lab. You will decide whether to accept or to reject. If reject, then the specimen need to be collected again. Rejection reason of rejection, maybe unlabeled or incorrectly labeled sample, leaking samples, outdated samples, etc. There are more reasons. Okay. You have any questions so far?
No.
Kelas ni tak ada yang lelaki kan? Nubhan seorang. Nubhan. Nabhan. Okay. Nabhan tak balik lagi. But you are here, Nabhan.
Ada.
Mana Nabhan? Ada. Nabhan seorang je kat sini ya.
Seorang lelaki.
Okay. It's not a problem, Nabhan. Nanti for the lab, I would like Shakira to get the students into groups, yeah, into groups. Group A and Group B. Group A berapa orang? Ada 29. So Group A tu 16 dan 13. 16, 13 ataupun 15, 14. 15, 14 15, 14. So Nabhan kena join lah mana-mana sisters punya group. You cannot be a group of your own. Okay? So right. Nanti bagi kat saya Shakira the list of name dalam group A. Group A need 4 smaller group, 1 group 4 person. Similarly for group B. So ada group nanti yang tak cukup members, dia ada 3 orang je, that's okay, but in general arrange yourself into group of 4. Okay, is that clear, Nabhan?
InsyaAllah clear.
Okay. Now just quick principles of the hematology lab. Okay, principle of hematology and immunology lab test. Firstly, good lab practice, and then medical knowledge, professionalism, patient-centered care, and then interpersonal and communication skills, and practice-based learning and improvement. So this must be always in your mind. Yeah, when we are in the lab, you must practice good lab practice, GLP. What are good lab practices? Firstly, samples must be treated as potentially hazardous. You must take safety precautions. You must know where to put the waste, how to keep records, how to maintain equipments, and use and implement SOP in hematology. So sekarang ni kena tambahlah SOP, COVID-19 SOP. Yeah. So that one is good lab practice. We'll talk about this in more detail later on, but this one is sepintas lalu je in brief. Firstly, good lab practice. Safety precaution involve wearing plastic apron, overlap coat, yeah, especially if you are handling a lot of hematology samples. For our lab, kita tak perlu pakai lah plastic apron. Tapi if you are in the hospital lab, yes, you have. Wear visor or glasses. Yeah. Mouth pipetting is forbidden at all time. You must clean up breakages immediately and use the appropriate cleaning materials like 10% hypochlorite. If breakage occurs in the centrifuge, pick up debris with a forceps, put bucket in the basin with 10% of hypochlorite, and leave overnight. Okay. At the end of the day, surface must be swabbed with 10% hypochlorite, meaning that you need to clean your area. Nanti masa masuk dalam lab saya, I will tell you lah how to create a working area and then a waste area. You will see that many people do not do this. Tapi saya buat. Okay, saya akan make sure you do it, and I will want to see that being implemented. Waste management will tell you where to put your rubbish in. Okay, medical knowledge. Okay, everyone who is in the lab must not do away with medical knowledge. You must develop, maintain a knowledge base in automated testing, peripheral blood analysis, although you'll be using the automated machine, etc. But we will expect you to know the concept, the SOP, all the related techniques, yeah, of using the materials and the machines. Examination and analysis of blood smears, analysis and interpretation of coagulation, thrombosis testing. We may not do this, but in real hospital setting, we will want you to know. Understand various levels of medical evidence and their translation into evidence-based practice. Okay. Professionalism. Okay, you must demonstrate compassion either to patient, family, other staffs, as well as physicians caring for them. Physician tersilap, doctors lah tersilap tick dekat form. Janganlah pergi marah pula. You must be compassionate. Tell them that you need to redo the blood test, tapi tak perlu marah-marah. Okay. And then positive work habits, punctuality, dependability, professional appearance. You must be responsive to patient and society, superseding self-interest. Okay. Confidentiality, tak ambil gambar yang bawa ke sana kemari. Tak perform a procedure without consent. You must be committed to excellence and ongoing professional development. Must also have interpersonal skills in functioning as a member of a multidisciplinary team, and above all must interact and act without discrimination. Jangan pula ini India punya apa tu make comments ke, no. Okay, there's no such thing when we do work in the lab. Okay, that's why dalam situasi perang sekalipun, we treat everyone, whether it is enemy or not. Yeah, we save lives. Okay, patient care. For patient-centered care, meaning that we must gather essential and accurate information of patient, effectively examine and interpret biopsies, aspirate cell counts and molecular cytogenetic information, effectively consult on interpretation of tests. Kalau tak sure, ask your supervisor, ask the seniors, consult the doctors. Effectively participate in the multidisciplinary conferences or meetings. So kat UK dulu memang every day I see that happen in the walk round. Also they have a specific multidisciplinary meeting. Everyone is there, the doctors, the lab people, the pharmacology, the pharmacy, the diet, nutrition pun ada. Even the social service worker pun will be in as part of the multidisciplinary team. I think it is becoming the norm in Malaysia these days. Interpersonal and communication skill covers things like being able to articulate the results, being able to write properly, okay, in a comprehensive and concise manner, including the history, the test done, the results, and also what is the analysis mean? What does the analysis mean? And then we can also offer suggestion, recommendation of what to do next. Practice-based, demonstrate knowledge of evidence-based medicine and apply its principle, develop personally effective strategies using lab problem to identify process improvement, and one last part ni, facilitate the learning of others. Bila kita ada student, we are open to student to come, we are open to doctors and also specialists to learn on the lab part of the service. Okay. Okay, last but not least, the hematology diagnostic market. We are talking about the need for the market for people who are trained in the hematology lab is huge. This is taken from the US data. It's similar in other part of the world. The need for a hematology diagnostic increased exponentially from 2014 and expected to keep on increasing up to years beyond 2021. Kat sini projection up to 2025. So this is in hospital, diagnostic lab increase. So diagnostic lab macam COVID kan, bukan semua hospital sahaja yang terlibat in the diagnosis of COVID-19, but also diagnostic labs. Diagnostic lab ni macam mana? Contohnya apa ni? Pantai, medical labs, granule labs, granule lab buat benda lain. Apa tu? Allah, lupa a few. But you need to start to know about the diagnostic lab that are available, because if you want to work, it's not only hospital that can provide you with some relevant work, but also diagnostic labs, independent labs. I have one master student degree in biotech. Bila dia habis degree, dia habis masters, terus dapat kerja dekat diagnostic lab. So dia walaupun dia biotech, okay, because the equipment is the same, the principle is the same. And she told me that the work is quite heavy during the COVID-19. Dia sampai tak ada masa nak rehat sangat lah, because bila kes sangat banyak dekat Selangor, she has to work overtime even. Okay. Others ni contoh macam research lab. So you see the hospital and the diagnostic lab, by 2025 they are expected to be equivalent in terms of the amount of work, amount of load, amount of demand for them. So nak kata apa, jangan risau tentang kerja, kena risau, but the job is there. You need to find, you need to mungkin for some situation you need to work a little bit harder and not just limiting your application to hospitals only. Okay. In terms of hematology analyzers and reagents, tadi tu macam the technical part. Ini analyzer and reagent. So there is increasing amount of demand for the analyzers and also reagent untuk pakai. So this is another thing that you may want to work in, either as part of the producing company or being a researcher for the machines and also for the reagent. Okay. In summary, hematology and immunology lab service is wide-ranging and requires the expertise to conduct many tests and equipments, either manual or automated. Personnel providing hematological service must also be equipped with personal soft skills and awareness of the purpose and function of the service. I open for any question if you want to ask. Raudah, ada nak tanya apa-apa?
Buat masa ni tak ada lagi, Doktor.
Okay. Nur Hayati, Nur Farhana, any question?
Tak ada lagi, Doktor. Buat masa ni.
So can we end our session. Siapa tu? Masturi. Oh, 28 bulan ni. Alamak. Okey. Siapa yang belum balik? Masturi. Okey. Belum balik tu. Nabhan dengan? Tak apa, saya ada nama. Thank you for reminding me about it. Tapi bila Nabhan balik, Nabhan does not have to be quarantined anymore, right?
Yes, ada.
Tak perlu kan?
Betul. Tak perlu.
Yang seorang lagi pun tak perlu quarantine apa dah kan? Maknanya you masuk ke campus, basically I can do the lab session. Tak perlu tunggu awak habis quarantine ke kan? Okay. I will have to reschedule. I will have to relook at the timetable. Okay. Saya tak boleh nak cakap apa-apa lagi sekarang. I will reschedule. Right? Okay, kalau tak ada apa-apa, that's all.
Doktor, saya nak tanya pasal grouping tu.
Siapa tu?
Syakira.
Syakira. Okay, yes.
Tentang grouping tu kan ada 2 grup, satu 15, satu 14. Kalau yang untuk 15, macam doktor kata buat group of 4 kan. So ada 4, 4, 4, 3 untuk grup A.
Okay, sekejap. Dia macam ni. Untuk lab tu, because kita kita ada 4 arm. Ada 4 arm untuk phlebotomy. So what we will do is satu grup akan buat phlebotomy dulu. Pada masa yang sama, another group will be doing the blood smear. Okay. In the next session, we will swap. The group that did the blood smear, they do the phlebotomy. The phlebotomy group will do the blood smear. They will run concurrently. Different time lah. Faham ke? Okay. Sekarang ni limitation kita pada phlebotomy group ni. Satu arm, we can have 4 people. So in total, we can have at one time 16 orang, 16 people during the phlebotomy session. Jadi akan ada 4, 4, 4 and 3. 4, 4, 4, 3. Yang grup satu lagi ni ada 14 orang kan. Okay. Bila dia swap nanti, grup yang kedua ni dia akan ada 4, 4, 3, 3 lah. Betul tak? 4, 4, 3, 3. Okay.
Sorry, kata 4, 4, 3, 3.
4, 4, 4. Grup yang pertama tu
4, 4, 4, 3.
4, 4, 3. Okay, grup kedua 4, 4, 3, 3.
Okay.
So in that case, any of your friends from the from the research pathway, kan? Research pathway. Actually, I can take group yang satu tu kalau dia orang nak masuk lah. Kita boleh accept one student to join. Lepas tu bila yang group kedua tu buat phlebotomy, kita boleh accept 2 student ataupun masa dia nak buat blood smear. I don't mind for them to join if they have the time, they want to join. I don't mind. Cuma dia tak ada assessment pada dia dan dia priority is for the actual student lah. Priority. Dia boleh join, cuma kalau apa-apa jadi, priority is for the actual student of that course. Cuma dia kena bagi tahu kat saya awal kalau dia nak join. Okay?
Okay.
Regarding OSPI, how we conduct OSPI, etc., closer to the date, I will tell you how do we organize. How do we organize? Tapi in general, Group A will do it first, Group B would be quarantine in a somewhere lah. It will be quarantine somewhere. Bila dah group A tu selesai, group A pula kena quarantine. Tunggu sampai group B selesai OSP. Okay. Dia akan jadi lama lah because at the same time you have a third group, the fourth year and the year 4 sekarang ni, they will also ni. So your quarantine period tu actually lama lah. Satu session at least 2 hours. So 2 hours of OSP, kemudian maybe up to 4 hours of quarantine. Okay. So nanti kita akan bincang lagi lah macam mana kita nak buat OSP kita. We also need to see whether we have finished our lab or not, and also whether there is MCO or not at that time. Hopefully there is none. Priority for OSP at the moment is for the year 4. Year 4 macam mana pun dia orang yang akan dibuat dahululah OSP for them. Okay, because you guys kalau jadi apa-apa, you still have a year to do this. You can do it next sem, but for them dah tak ada masa. Okay, alright. Kalau tak ada apa-apa, ada apa-apa lagi tak?
Tak ada.
Semua dah sign in attendance You have until Sunday to do that. Okay. Okay. Wassallallahu ala sayyidina Muhammad wa ala alihi wa sahbihi wa sallam walhamdulillahi rabbil alamin.
Thank you, Doktor.
Okay, thank you.